Good morning, sir. I'm NJ Lahera from BSN 1D. So for today's video, I'm going to discuss the glycolysis, what is the important and the step-by-step process of glycolysis and more. Glycolysis is a pathway that converts glucose C6H12O6 into pyruvic acid CH3COCOOH. The free energy released in the process is used to form the high-energy molecules adenosine triphosphate and reduce nicotinamide adenine dinucleotide. Glycolysis is a sequence of 10 reactions catalyzed by enzyme, a process in which glucose, sugar, is partially broken down by cell enzyme reaction that do not need oxygen. Glycolysis is a method that cells use to produce energy. Glycolysis, what is so important? Glycolysis is important in our cell because glucose is the main source of fuel for our tissue in our body. So we have two phases in a glycolysis. So the first phase is a preparative phase. Preparative phase is requires an input of energy in the form of 2 ATP per glucose molecules. So the second one is a energy generating phase. Energy generating phase is energy is released in the form of 4 ATP 2 per glyceraldehyde molecules. The first step is a glycolysis conversion of D-glucose into glucose 6-phosphate. The enzyme that catalyzes this reaction is hexokinase. So the second reaction of glycolysis is the rearrangement of glucose 6-phosphate into fructose 6-phosphate by glucose phosphate isomerase or phosphoglucose isomerase. The third reaction is phosphorylation reaction that therefore requires expenditure of energy. The enzyme is involved in here is phosphoproctokinase. The step 4 is cleavage, formation of 2-triose phosphate. The reacting C6 biphosphate is split into C3 monophosphate species. Because fructose 1,6 biphosphate, the molecule being split, is unsymmetrical, the 2 trioses produced are not identical. Step 5. Isomerization, formation of glyceraldehyde triphosphate. Glyceraldehyde triphosphate is a glycolysis intermediate. The hydroxyacetone phosphate in the other triose can be readily converted into glyceraldehyde triphosphate. The enzyme triose phosphate isomerase catalyzes the reaction. So we are in the second phase, the generating energy phase. The step 5 is isomerization, formation of glyceraldehyde triphosphate. Step 6, oxidation and phosphorylation using phosphate, formation of 1,3-biphosphoglycerate. So glyceraldehyde triphosphate, the hydrogenase, is added an organic phosphate to glyceraldehyde triphosphate, producing 1,3-biphosphoglycerate. So in this step, two main events that take place. So glyceraldehyde triphosphate is oxidized by the coenzyme nicotinamide adenine dinucleotide. The molecules is phosphorylated by the addition of free phosphate group. The enzyme that catalyzes this reaction is glyceraldehyde triphosphate dehydrogenase. Step 7. Phosphorylation of ADP, formation of 3-phosphoglycerate. Diphosphate is converted back to monophosphate. This is an ATP producing step 1. which the C1 phosphate group of 1,3-biphosphoglizerate high-energy phosphate is transferred to an ADP molecule to form ATP. The enzyme involved is phosphoglycerokinase. ATP production in this step involves substrate-level phosphorylation. It is a process whereby ATP is produced from ADP by hydrolysis of high-energy compound. Step 8. Isomerization. Formation of 2-phosphoglycerate. This step involves a simple rearrangement of position of the phosphate group of 3-phosphoglycerate molecule, making it 2-phosphoglycerate. The molecule responsible for catalyzing. The molecule responsible for catalyzing this reaction is called phosphoglycerate motifs, PGM. A motif is an enzyme that catalyzes the transfer of functional group from one position on a molecule to another. Step 9. Dehydration. Formation of phosphonol pyruvate. This step involves the conversion of 2-phosphoglycerate to phosphonol pyruvate. The reaction is catalyzed by the enzyme enolase. Enolase works by removing a water group or dehydrating the 2-phosphoglycerate. The specificity of the enzyme package allows for the reaction occurs through a series of steps, too complicated to cover here. Step 10. Phosphorylation of ADP, formation of pyruvate. So, this is the final step of glycolysis. Convert phosphonol pyruvate into pyruvate with the help of enzyme pyruvate kinase. So, this reaction involves the transfer of phosphate group. The phosphate group attached to the carbon 2 of PEP is transfer of molecule of ADP yielding ATP. Again, since this is the 2 molecule of PEP, here we actually generate 2 ATP molecules. Phosphonol pyruvate transfer its high-energy phosphate group to an ADP molecule to produce ATP and pyruvate. The enzyme involved is pyruvate kinase.
1, 2, 3